Good morning, students. We will continue lipid metabolism. Today's competency is competency number 4.3, and in this we are studying mainly the metabolism of lipoproteins. First, we will study chylomicron metabolism. These chylomicrons are synthesized by the small intestine after meal. When these chylomicrons are synthesized, they mainly consist of triglycerides and cholesterol esters in the core, and it has two coverings of apoproteins, that is ApoB48 and ApoA. Now when this type of chylomicron, which is synthesized in the small intestine and enter into the blood, it is called as nascent chylomicron. In the blood, the HDL lipoprotein gives two more covering to this nascent chylomicron, and they are ApoC and ApoE. Due to addition of these two covering, now nascent chylomicron is converted into mature chylomicron. Now when this mature chylomicron goes to the peripheral tissues, and when it is traveling through the capillaries of the peripheral tissues, then the enzyme called as lipoprotein lipase, which is present on the capillaries of peripheral tissues, this enzyme is activated by a covering of chylomicron, that is ApoC. After activation, this lipoprotein lipase acts on the triglycerides present in the chylomicron, break these triglycerides into glycerol and fatty acids. Fatty acids are utilized by the peripheral tissues, that is extrahepatic tissues, and this glycerol is transported to the liver, because for utilization of glycerol, the enzyme glycerol kinase is required, which is only found in the liver. Now when this large amount of triglycerides is hydrolyzed by lipoprotein lipase, then the size of chylomicron decreases. At the same time, this chylomicron loses its two covering, that is ApoA and ApoE. ApoA and ApoC is lost. Now only two coverings are remaining, that is ApoB48 and ApoE. Now such chylomicron is called as chylomicron remnant. Now this chylomicron remnant is transported to the liver. In the liver, there are two types of receptors are there, LDL receptors and LRP receptors. LDL receptors, long form low density lipoprotein receptors, and LRP receptor means low density lipoprotein related proteins. So these two receptors takes the chylomicron remnant, and then inside the liver, the small amount of triglyceride and cholesterol which is present there is utilized. So in this way, the chylomicrons are metabolized in the body. Now we will study VLDL metabolism, very low density lipoprotein metabolism. Now VLDL is mainly synthesized by liver to transport the lipids which are synthesized by the liver. So the liver will synthesize mainly the two types of proteins, that is triglycerides and cholesterol esters. So these two types of proteins are added into the VLDL. At the same time, the two coverings, that is ApoA and ApoB100, are added to it and released into the blood by the liver. Now this VLDL is called as nascent VLDL. When this nascent VLDL enters to the blood, the HDL will donate two more covering, that is ApoC and ApoE, to it. Now this VLDL is now covered by four types of coverings: ApoA, ApoB100, ApoC and ApoE. Now this is called as mature VLDL. Now when this mature VLDL goes to the peripheral tissues, just like chylomicron, the lipoprotein lipase enzyme is activated with the help of the covering of VLDL, that is ApoC. After activation, this lipoprotein lipase acts on the triglyceride present inside the VLDL, break the triglyceride into free fatty acids and glycerol. So they will come out of the VLDL molecule. Fatty acids are utilized by the peripheral tissue. Again, the glycerol is transported to the liver for its utilization. When large amount of triglycerides are hydrolyzed by the lipoprotein lipase, then small amount of triglyceride will remain. Then its size get decreased and it is called as VLDL remnant. It has only two coverings, that is ApoA and ApoC is lost. Only two coverings are there, that is ApoB100 and ApoE. Now this VLDL remnant is also called as IDL, intermediate density lipoproteins. Now through its journey, this VLDL remnant loses more triglycerides to the peripheral tissue, as well as a covering, that is ApoE, is also lost, and it get converted into low density lipoprotein. Here the density will slightly increases because of loss of large amount of lipids to the peripheral tissue. Therefore VLDL is converted into LDL, low density lipoprotein. So this low density lipoprotein has only one covering, that is ApoB100, and the main lipid which is present in the LDL is cholesterol esters. Now this cholesterol is, this LDL is taken up by three tissues. First, by extrahepatic tissues. So these extrahepatic tissues take the LDL through the receptors present on the extrahepatic tissues. They are called as LDL receptors. Liver also take this LDL back with the help of LDL receptors. The third fate of LDL is they are also engulfed by macrophages. This uptake of LDL by macrophage is not dependent on receptors. Therefore, these macrophages will take large amount of LDL when its level is increases in the blood, and they get converted into foam cell. So in this way there is metabolism of VLDL. Now coming to the metabolism of HDL, high density lipoprotein. This high density lipoprotein is synthesized by liver and small intestine. When it is synthesized, it does not contain any lipids inside. Only it has that outer core which is made up of phospholipids and free cholesterol, and there is a covering of protein called as ApoA. Now in this outer covering of phospholipid, a enzyme called LCAT is present. The long form of LCAT is lecithin cholesterol acyl transferase. Now this HDL is enter into the blood. It is also called as discoid HDL because it is flat, because the inner core is empty. So it is flat. Only the phospholipid covering is there and ApoE protein covering is there, along with the enzyme LCAT in the membrane. Now when this discoid HDL goes to the peripheral tissues, then in the peripheral tissues some receptors are present on the extrahepatic tissues. They are called as ABC1 receptors. Long form is ATP binding transporter or cholesterol efflux regulatory proteins. So these receptors take the cholesterol from the peripheral tissues and transport it to the HDL. Now this cholesterol is free cholesterol. Now when the free cholesterol from peripheral tissue is transported to the HDL, this free cholesterol does not enter in the inner core. Therefore the enzyme LCAT convert the free cholesterol into cholesterol esters, that is hydrophobic proteins, lipids, sorry. and therefore after conversion of cholesterol into cholesterol ester, they get occupied the empty space of the inner core of HDL molecule. So in this way the HDL is added. Now this is the diagrammatic representation of conversion of cholesterol to free cholesterol. On the upper slide I have shown you free cholesterol, which is obtained from the peripheral tissue. Now the enzyme lecithin cholesterol alkyl transferase will transfer a polyunsaturated fatty acid, and this polyunsaturated fatty acid is present in the phospholipid covering of HDL. Now so polyunsaturated fatty acid is transferred to the free cholesterol, OH group of free cholesterol, and that free cholesterol is converted into cholesterol ester. When this free cholesterol is converted into cholesterol ester, then it easily goes to the empty core of the HDL molecule. So this is the role of LCAT is very important. At the same time, the presence of polyunsaturated fatty acid in the phospholipid, mainly lecithin contains this polyunsaturated fatty acid, so is important for transport or conversion of free cholesterol into cholesterol ester. If free cholesterol is not converted into cholesterol ester, then it does not goes into the core of the HDL molecule. So in this way the cholesterol is taken up, it gets converted into cholesterol ester, and then it is added to the inner core of the HDL molecule. Therefore the size of HDL molecule increases, it bulges. Now it becomes a spherical shape. Initially when it is synthesized, it has discoid shape. Due to deposition of cholesterol esters in the inner core, its size, it bulges and its shape changes to spherical. Now when it is fully filled then with cholesterol ester, then it is called as mature HDL. Now when this mature HDL goes to the liver, it is taken up by the liver with the help of SRB1 receptors. The long form of SRB1 receptor is scavenger receptors, class B type 1. So with the help of this receptor, the liver take the mature HDL molecule, and the cholesterol ester which is present inside that HDL is converted into neutral sterol, and it is excreted through the stool. So in this way there is metabolism of lipoproteins takes place in the body. Thank you very much.